All right, I'd next like to welcome Dr. Bill Gibson for his presentation on rare disease variants informing common disease risks. Dr. Gibson is a clinical geneticist with an interest in overgrowth, severe obesity, and lipodystrophy. His group applies state-of-the-art assessment of body fat mass, fat distribution, and circulating hormones to individuals with overgrowth, severe obesity, and genetic lipodystrophy syndromes. In collaboration with the British Columbia Genome Sciences Center, the group combines the detailed metabolic profile with targeted assessment of copy number variants and specific mutations in an effort to discover the cause of the condition. Ultimately, their goal is to design therapies for these rare disorders. They then hope to translate these discoveries into viable treatments for prevention of obesity, type 2 diabetes, brain aneurysms, and cardiovascular disease in the population as a whole. So please welcome Dr. Gibson.
Marvelous. Thanks very much, everyone. It's a pleasure to be speaking here at the TBRS Summit. It's become traditional for Canadian academics to offer a land acknowledgement at the beginning of the talks. So I'd like to acknowledge that San Antonio is located on the traditional and ancestral territory of the Payaya people of the Tatpilam-Kwawiiltekan Nation, and that UBC is located on the traditional, ancestral, and unceded territories of the Coast Salish peoples, the Musqueam, Tsleil-Waututh, Squamish, and Stola First Nations. So I'll be talking about how we can use studies of rare disease variants to inform our understanding of risks for common disease. Now, I think for most members of this audience, it's very clear about how DNA is wrapped around histone proteins and how epigenetic modifications, including DNA methylation, histone methylation, various other post-translational modifications on histone proteins influence gene expression and influence the life course. I think of it, and I describe it as a kind of a soft wiring of the DNA sequence, with the actual DNA code itself being the hard wiring, and epigenetic modifications, opening and closing of chromatin as a kind of a soft wiring. So my group focuses on the polycomb repressive complex. This is a cartoon diagram of that here, which has three core components, EZH2, EED, and SUS12. Let's see, is my... Whoops, what am I doing there? Is this the pointer? Well, you can see it over there on the left of the screen. A core components made up of the three main members with a series of other components that swap in and out for various processes. Now, this is a diagram of the complex bound to DNA itself. The EZH2 and SUS12 components seem to provide the catalytic activity, whereas EED provides the reader function. So there's epigenetic readers, writers, erasers, and the EED moiety provides the reader function. So there is some segregation of function there. Now, the clinical phenotype of the PRC2 complex disorders it overlaps quite strongly with Tatten-Brown-Rahman syndrome. So there's overgrowth, intellectual disability, developmental delays, some characteristic facial features, as well as early maturation of the bone age, and a variety of skeletal malformations as well. This is from some of the work of the Tatten-Brown team with a series of facial photographs of patients with Weaver syndrome. You can see some of the facial features here, widely spaced eyes, tall forehead, prominent forehead, also a chin that seems sometimes to be receding, sometimes what we call retrognathia there, broad nasal root, broad nasal bridge. Now, rare variants in EED, which is the reader component of the PRC2 complex, also cause its own unique syndrome, shown here. Again, here are some of Kate's patients, as well as one of ours, with facial features that very distinctly resemble those of Weaver syndrome, though you can see one of the patient on the upper, to the left of the screen there, but on the upper right, in adulthood really doesn't have a receding jaw, but her jaw is quite prominent. Now, After my PhD student Anna Cohen and I had described Cohen-Gibson syndrome, my new PhD student, Shari Cyrus, who's a physician from Barbados, went on to collect the patients that we had in our own registry of rare PRC2 complex disorders who had missense variants and frame shift variants in the SUS12 component. So we were able to refine the phenotype of Imagawa-Matsumoto syndrome with a case series of our own. Again, you can see the patient in the sort of upper left there has facial features quite similar to Weaver syndrome, though not every patient has that overall facial gestalt. So the facial gestalt does seem to be more variable for Imagawa-Matsumoto syndrome. Again, the overgrowth can be variable. We did see a larger number of cardiac abnormalities in patients with mutations in SUS12, and we think we saw a greater prevalence of structural brain malformations as well. Kate has talked about the mutational spectrum of different disorders. You can see here EZH2, EED, and SUS12. A wide variety of missense variants in EZH2, though again, the truncating variants seeming to be concentrated in the last exon there. Predominantly missense or in-frame indel variants in EED, but a greater proportion of frame shift variants in SUS12. Now, we went on to try to see if we could make an assay that would tell us the relative difference in function between some of the variants that we were seeing in patients with EZH2 and EED. Anna Cohen, working with Damien Yap at the BC Cancer Agency, was able to design an assay that looked at a substrate G9A and found that rare variants in EZH2 that had been associated with Weaver syndrome reliably clustered as perhaps not complete loss of function mutations, but certainly significant loss of function mutations. The kicker, though, was that when we built in a variant that was known to be a benign variant, that variant also segregated with the Weaver syndrome variants. So it's, I can't really use that assay to tell the difference between benign variants and pathogenic variants. But it is important to be able to do so. What I'm showing here are two growth curves of two different patients with different missense variants in EZH2. On the left is a gentleman who doesn't have intellectual disability, though he is a mosaic, thought to be a mosaic for this mutation, but he doesn't have intellectual disability, but clearly does have tall stature and high body weight, whereas the patient on the right of the screen has microcephalic primordial dwarfism, but also has a missense variant in EZH2. She effectively has the reciprocal or opposite phenotype of Weaver syndrome, at least from a growth perspective. So we contracted with a private company called BPS Bioscience to do a bioluminescence assay driven by that specific variant and also by the wild type. And you can see here that that alanine 738 to threonine variant, in fact, had increased methyltransferase activity. What you've got on the left of the screen is a heat map showing the different epigenome-wide profiles produced by Rosanna Wexberg's lab. And to the far left are patients with Weaver syndrome, so blue on the top, yellow on the bottom. In the middle are patients without Weaver syndrome, so that's basically the normal controls. But you can see there on the far right with the bright yellow on the top and a little bit of blue and yellow on the bottom, that's that patient with the constitutively activating mutation. So in fact, not only is her mutation activating the methyltransferase activity, but the genome-wide, epigenome-wide profile is in fact the reciprocal of what one would expect with Weaver syndrome. By that I mean that areas of DNA that are reliably more methylated than average in Weaver syndrome are reliably less methylated in this patient, and the reverse is true. Areas that are reliably less methylated in Weaver syndrome are more methylated in this patient. So the epigenome-wide profile helps us discern some of the functional effects downstream of this. Now, a vexing problem that many of us are well familiar with is what to do about all these rare variants that we see in NOMAD. Now, for those of you who don't look at NOMAD as often as I do, so NOMAD is a database of about 150,000 adults, largely recruited from NIH studies. They're not necessarily certified as healthy, but the patient set is believed to be relatively depleted of individuals with childhood onset neurodevelopmental disorder. So for a lot of neurodevelopmental conditions, NOMAD is used as our quote-unquote normal controls. But clearly, with a large number of missense variants in there, all of these individuals can't all have Weaver syndrome. Now, they may all or many of them have clonal hematopoiesis as an explanation for why these missense variants are found in their blood, because these are largely blood-derived DNA samples. We'll talk a little bit about that later on in the talk. We did also see that this observation holds true not just for EZH2, but also for EED, and in fact for every rare Mendelian disorder gene that I've checked. So if it's quote-unquote an OMIM gene, if it's a gene that when mutated causes a pediatric neurodevelopmental syndrome, there are multiple rare missense and nonsense variants in NOMAD for all of the genes that we as clinical geneticists pay attention to. So one of the things that we know is that these variants are typically classified as variants of unknown significance until they're annotated specifically to a rare genetic disorder. What I'm showing here are sections from the classification criteria that the American College of Medical Geneticists and Association of Molecular Pathologists use, just to make the point that what I've drawn in the boxes there, a well-designed functional assay can help us make or break the call between a variant of unknown significance and a likely pathogenic or a likely benign variant. Now, we also use the ACGS criteria by the Association of Clinical Genomic Science in the UK. This has been pitched as basically a temperature graph, so you can see at the very top there's pathogenic variants, at the very bottom there's benign variants, common in the general population. But the ACGS criteria were designed as a way to help us relatively categorize variants of unknown significance. So not just treating it as one big bin, but saying, well, these ones look a lot more suspicious than others. So that's semi-colloquially, there's hot VUS, warm VUS, tepid VUS, and sort of somewhat chillier VUS. And actually, our own molecular lab at BC Children's Hospital uses this to decide which VUS to actually report out and which ones not to. So, again, I've mentioned to you that Anna Cohen and I had tried to get an in vitro, so cell-free assay to work, but we just weren't confident that it was reliable. If it can't tell the difference between a known benign variant and a pathogenic variant, we probably shouldn't be using it to make those calls. So we'll be hearing a little bit about mice. We've certainly heard about clinical phenotyping in humans. I'm not only going to switch gears, but I'm going to, in fact, get into a completely different vehicle and talk about fruit flies now. So one of the things that's known is that the polycomb repressive complex, the PRC2 complex, the complex that when mutated causes these three syndromes that I've talked about here, it was actually discovered first in fruit flies. And the core complex is very well conserved between humans and flies. So I know it seems counterintuitive to be using fruit flies in a situation like this, but the fact of the matter is that these are very, very old proteins, and they still do pretty much the same thing in humans as they do in flies. So the complex has essentially the same structure. There's a variety of slightly different names for things. So EZH2 is called EZ, EED is called ESC, SUS12 also called SUS12, a variety of different names for the other components that are present in the fruit flies. So what we did then is we looked, because we were interested in solving ways of classifying rare variants, be they variants of unknown significance or known pathogenic variants, we lined up the human EED protein with the fly ESC protein and sought to say, hey, you know, we know that these variants that cause Cohen-Gibson syndrome seem to be occurring in variants that are conserved between humans and flies. We've got some known benign variants from NOMAD. Let's see what we can design there. So we set up to put together what we call fly mimetic assays. In other words, we're taking the site that is at which the mutation occurs, mutating it in the fly version of the gene and seeing what happens in live fruit flies. So this is a schematic of where we've made these variants. We've taken some likely benign variants from Nomad, some known pathogenic and likely pathogenic variants from the literature, some known pathogenic variants from hematological malignancies, so not necessarily constitutional variants that children are born with, but in fact that have developed as a somatic mutation throughout life, as well as some variants of unknown significance. We've built these into a construct. This is, by we, I really mean my collaborator Douglas Allen, who's a very talented Drosophila geneticist at the University of British Columbia and already is very successful in assaying P10 variants in Drosophila assays. I was lucky enough to go to one of his talks at the Children's Hospital Research Institute thinking, hey, this would probably work with PRC2 complex. And I mean, spoiler alert, it kind of does, which is very pleasing. So what I'm showing here is the construct that we've basically built into the flies. Effectively, there's the fly ESC gene built in a construct that has a swappable cassette in there. So the ordinary transcription will result in a truncated protein, but will then immediately activate the cDNA for our mutated protein, and that allows us to swap in different missense variants and test them. So a little brief digression into fly physiology. Essentially, fruit flies have, male fruit flies are supposed to have only 2 sex combs. I see I'm getting the 5-minute warning, which is a bit strange because I think don't I have a half-hour talk? But anyway, so the thing to know is that if they have more than 2 sex combs, that is indicating that the mutation is having an effect. So it's indicating partial loss of function of the PRC2 complex. So what we see here is in our extra sex combs assay, the appearance of sex combs on limbs other than the foreleg counts as extra sex combs. That's in fact how the gene itself was named, ESC for extra sex combs. So this is the work of Shari Cyrus, again, the physician from Barbados who's recently got her PhD from my lab. I'll just walk you through this sort of color scheme that we have. Essentially, on the far left, the green bars are when we built the constructs into flies and counted the male flies. If there's only 2 sex combs, that's a green bar. So in the lower right there, you'll see that 2 sex combs is a green bar. Anything more than 2 is either lighter green, yellow, orange, or red. So what you can see is that when we calibrated our assay, the pathogenic variants from Cohen-Gibson syndrome all reliably caused extra sex combs to appear in the flies. Now, we then did what's called a replication set where we put other pathogenic variants in there. And again, each of these variants derived from Cohen-Gibson syndrome patients, including an in-frame indel, generated extra sex combs in the flies. So we're able to correctly discern pathogenic and likely pathogenic variants from wild type and from likely benign variants. We were also able to show that variants derived from hematological malignancies have partial loss of function and loss of function effects, and in fact build in some variants of unknown significance, showing that 3 of these would be predicted to be classified as benign, whereas 1 of them would actually be predicted as pathogenic. I should mention that last variant has come from a patient who had the clinical phenotype of Cohen-Gibson syndrome, but at least in the paper itself, it was classified as a variant of unknown significance because it couldn't be, the parental segregation could not be confirmed. De novo status could not be confirmed. We also took that in-frame indel and broke it apart into its 2 component parts and showed that you can see the far left with the change of the 2 amino acids, it appeared to be pathogenic, whereas if we made only the N-terminal change, it was predicted to be benign, and it looks like the amino acid change, basically the second half, is the disease causing, is the one that is interfering with the protein's properties there. So we now believe that we have a Drosophila assay that can accurately classify variants of unknown significance in EED, at least for variants that are occurring in amino acid residues that are shared between humans and flies. What's next? Well, we're moving on to then checking different variants in SUS-12. And using a similar approach with a variety of different missense variants. And so the question then becomes, well, how could reclassifying variants from the NOMAD database be useful in understanding how these rare variants might contribute to common complex disease? I mean, it's important to know that many of the NOMAD participants do in fact have common complex diseases: diabetes, cardiovascular disease, neuropsychiatric phenotypes, etc. The prevalence of these missense, in-frame indels, and nonsense variants is non-trivial. When I added it up across these different genes, you can see about 1 in 400 NOMAD participants have a rare variant in EZH2, about the same in EED, and even more common in SUS-12 or some of the other components. So adding up across the entire PRC2 complex, about 1 in 44 NOMAD variants, or 1 in 44 NOMAD participants has a rare variant in this complex. So are these likely to be constitutional variants or clonal variants? Hard to say. For clonal hematopoiesis, many researchers have what they call the variant allele fraction, and that's anything above 2%. Of course, in the curation of the NOMAD data, these are all individuals who are called as essentially fully heterozygous. So we imagine that some of them would have clonal hematopoiesis, but many of them may also may instead have these as constitutional variants that probably aren't causing full neurodevelopmental syndromes, but may be contributing to their health. So since our assay can classify not only variants derived from hematological malignancies, but also constitutional variants, we're confident that it can be useful for things like rare variant burden studies that can help us test associations with rare variants in these genes and common complex phenotypes. So we're moving on with these to establish other assays for EZH2 and SUS-12. We also have a longitudinal registry for patients with rare variants in PRC2 complex and other epigenetic disorders in British Columbia and elsewhere. And I'd like to acknowledge the strong contributions from the students who have worked in my lab and also our collaborators at Douglas Allen's lab, as well as the referring physicians and our funding agencies. Thanks very much. And I'm happy to take questions from the audience.